Hello, welcome to our virtual CU Anschutz alumni series, Happy and Healthy Hour. We are grateful that you are here with us today to connect with the CU Anschutz alumni community and to learn from our amazing experts here at CU Anschutz Medical Campus. My name is Kara Padgett, and I'm the Senior Director of CU Anschutz Alumni. Our office has numerous ways for all alumni to get involved with the campus. Our alumni team is always happy to help connect you with alumni activities for your school or college. Keep an eye out for an email after this evening's event where we will share upcoming events and how to reach the team. Before I introduce tonight's speaker, I want to draw your attention to the Q&A function at the bottom of your screen. While we have some time designated at the end of the presentation for Q&A, you are also welcome to enter a question at any time using the Q&A function. Additionally, we have alumni team members available on the chat if you need anything during tonight's presentation. I now have the privilege of introducing tonight's speaker. Dr. Aimee Bernard is a PhD immunologist with specific expertise in human B cell development and the autoimmune disease SLE. She is also the executive director of the CU Human Immunology and Immunotherapy Initiative. As an educator, she shares her knowledge of immunology with medical, dental, physician assistant, and graduate students from across the CU Anschutz Medical Campus. Aimee is a passionate pro-science advocate with over 10 years experience testifying in support of common sense public health legislation. Her current medical education research focuses on the convergence of science education and science communication and aims to empower scientists and healthcare professionals to communicate more effectively with the general public. Please join me in welcoming Dr. Bernard.
There we go. Hi, thank you so much. Thank you for having me. It's my pleasure to be speaking to you tonight about vaccines and the immune response. So I'm going to share my screen here. Yeah, and as Kara mentioned, I'm an immunologist here on campus, and my main role as an immunologist is I'm an educator. So I teach pretty much all of the students across campus here. So med students, dental students, graduate students, physician assistant students. I'm going to teach in a couple of different of the graduate programs. So immunology is my passion, and I have chosen to teach others about it for a living. So tonight we're going to talk about your shot at protection and how vaccines train the immune system to fight disease. And before we get started, I love when people have questions. So it's my understanding that you can't ask anything during the webinar, but please type your questions in the chat, and I'd love to have a conversation with you when the webinar has ended. We can have a nice discussion. So make sure you add those questions in a timely fashion so you don't forget. Okay, so let's get started. So these are the questions that we're going to cover tonight. So what is the immune system? How does it work? What is a vaccine? How do they work? Why are there so many different formulations of vaccines? And then finally, why should I get vaccinated? So we'll cover all of those in pretty good depth. So first question I have for you, have you ever trained in the martial arts? And if you are not a martial artist, or if you have seen martial artists, they train in self-defense. So martial artists, their whole goal, it's not offense, it's defense. They want to defend themselves. So as you can see from the cover of this book, it's Bruce Lee, an amazing and famous martial artist. It's training in self-defense. So how do they do that? Well, first, they're going to study their enemy. Martial artists watch competitors. They watch their enemies, and they study how they fight. What are the things that they do most frequently? What are their techniques? What are their tactics? So martial artists will study their enemy so that they can learn from that. And they remember how their enemy fights. So martial artists, when they get into a fight or a competition with their competitor, they are going to remember how they fought or how they watched them fight, and they are going to defend themselves in the best way, use the techniques that are going to be most effective. As a martial artist myself, when you are in the ring, you want the fight to be over. So the best thing to do is to figure out how they fight and defeat them at their game. So why am I telling you this? This is not a webinar about martial arts. This is to tell you that immune cells are like martial artists. So I have now turned Bruce Lee into an immune cell, and I have turned his competitor into a virus. That is an awesome sidekick. Perfect execution. So immune cells, just like martial artists, will train in self-defense. And there are certain cells of the immune system that do this. And we'll talk a little bit more in detail in a couple of slides. But in general, we can say that immune cells train in self-defense. They study the enemy, whether it's coming in the wild, like you got it at the grocery store, or in vaccine form, as we'll talk about. You study the enemy and you remember how the enemy works. And this is making immune memory. So some cells of the immune system will create memory. These memory cells can patrol our body for our lifetime and protect us from invading pathogen, particularly those that they have seen and studied before. Oops, let's see. Yes, so immune cells are going to respond to every encounter with a pathogen to protect our body and to defeat the enemy. So anything that comes our way, our immune system is going to respond to everything that comes our way. I use the word pathogen. I know that you're all CU alumni. Most of you are likely very well versed in some of these terms, but I just want to make sure everyone's on the same page. But pathogens are those microscopic organisms. They invade our body. They try to take it over and make it their home. Viruses, bacteria, parasites. Turns out that our body is kind of an amazing place to live. Nutrients, moisture, heat, kind of a nice place to live. So our immune cells are going to respond to every encounter with a pathogen to make sure that the invaders stay out and our body is defended. Another thing I want you to know about the cells of the immune system, and this is a homemade graphic design slide. So I will not quit my job as an immunologist to go into graphic design, but I wanted to drive home the idea that the cells of the immune system are a mobile defense system. So just like first responders or an ambulance, immune cells will be called to the site of injury. They're going to travel to the site of injury. So you can see in this bottom right of the screen, there's a cut on the cartoon hand. You're going to see swelling, redness, heat, pain. These are all the result of the damage, but also the immune cells being called to the area. So you're probably familiar with inflammation. That's because immune cells, the mobile defense system, is going to be recruited to the area where it's needed, to that site of injury or damage. The immune system, it's a system and network of cells and some chemical mediators that work together. And immune cells do have certain organs that they reside in, but they are mobile. They're always moving through our blood system. So they don't have like just one specific location that they live. Like you think of your lungs or your heart, like that's that one organ where, the function of breathing or, pumping your blood occurs. The immune cells are throughout our entire body, and they're always moving and being recruited to the area where they're needed. So they're mobile, they're always moving. So a mobile defense system. There are a lot of different types of immune cells in our bodies. You've probably noticed by now that I'm a huge cartoon fan. I do this for medical students. I do this for dental students. I do this for PhD students. So cartoons, it's not necessarily to simplify it. It's because I find it entertaining, and I think it helps you to remember if you have a little bit of maybe humor or fun. So we have a lot of different types of immune cells in our bodies. This is not representing all of them, but it's a pretty good number. Important to know that they have slightly different jobs. So I thought I'd walk you through a couple of the cell types here. So let's look at this cartoon. I'm going to draw your attention to the macrophage, kind of like the bottom right, this big white cell. Just what it sounds like, it's a big eater. This is a cell that the way that it defends our body is it eats things. So it eats cellular debris, it eats pathogens, it could eat dead and dying cells. So the macrophages, I like to think of as like the garbage truck or the disposal of the immune system. So that's one cell type. Another cell type I would like to draw your attention to is the bottom left. It's DC. That stands for dendritic cell. As you'll notice, this one is also eating. So they will also ingest pathogen or particulate matter. They'll take it in to remove it. But the dendritic cell has a slightly advanced function. Dendritic cells will take in pathogen. They'll actually eat it. They'll process it, and they will actually then present or display bits of the broken down pathogen. It's peptide in particular, so small bits of protein from the pathogen. They'll display it on their surface. And the reason why they do that is they then show off what they found to T cells. So if you look at the top left, the helper T cell, and the top right, the killer T cell, a dendritic cell is thought of as a scout or a sentinel. You'll notice that I like to give military analogies for the immune system. It is our defense. The dendritic cells act as sentinels or scouts because they take in pathogen, they process and they present bits of peptide or small bits of protein from that pathogen on the surface of their body, and they go off and they find T cells and they show it off to T cells. So that is their role. They are alerting the T cells that an invader has come into the body and that they need T cell help to defend the body and to eliminate the pathogen. So let's talk about the helper T cell next. So if this helper T cell has been alerted by a dendritic cell of an invader, the helper T cell, its job, just like its name sounds like, is it helps other cells to become better at what they do. So helper T cells will help cytotoxic or killer T cells, the top right. They will also help the bottom right cell here that we'll talk about in a second, called a B cell. Their job is to help other cells become activated and become better at what they do, kind of have increase or enhance their job, the way that they work. So let's talk about the killer T cell next. So on the top right, this is another cell in our body, immune cell, and killer T cell is kind of its nickname. Its official name would be cytotoxic T lymphocyte, but I prefer to call it a killer T cell. Essentially what these cells do once they've been alerted by scouts or sentinels is that these cells will actually do the hand-to-hand combat of killing infected cells. So killer T cells, just what they sound like, is they kill the infected cells to defend the body. So they're kind of like the ultimate warrior. That's why this guy has on a ninja outfit and the sword. The last one I want to draw your attention to is the bottom right. This is a B cell. I am a B cell immunologist, so I might be a little bit biased. B does not stand for best, even though in my mind it does. But B cells are the cells of the immune system that produce antibody. And if you look at this drawing, you'll notice these kind of like Y-shaped protein molecules on the surface. That's antibody. So that's the protein that B cells will produce. And I like to think of B cells as the archer of the immune military, because they will produce and secrete or fling their protein arrows or antibodies into the blood. And they'll make a ton of antibody to go off into the blood. This antibody will bind to the pathogen, so like kind of coat the pathogen and render it ineffective. So we say neutralize. And that's why people often talk about, well, is there neutralizing antibody? Because if you coat something completely with antibody, like if you coat the virus completely, that virus cannot infect its target cell, like your lung. So the antibody will coat and neutralize. That's why it's so important that we have high levels of antibody in our blood in response to like the COVID vaccine, is we want that antibody in our blood to be able to coat the virus if it enters our body and render it ineffective. So to prevent that disease. Okay, so I haven't talked about all the cells, but you can see that we have multiple types of immune cells in our body, and they have slightly different jobs. Now, it's important to understand that they all work together to defend us against pathogens. So some kind of come on early, some take a little bit longer to become activated or join the fight. But all in all, the immune cells are going to be working together to defend us against pathogen. And they have some unique jobs, and they also have some overlapping jobs. But combined, all of these cell types help our immune system to function properly. So I want to tell you a little bit more about some of these cell types. So some immune cells we say are non-specific. And these are referred to as cells of the innate arm of the immune system. So what does that mean? Well, that means that if an innate cell, like a macrophage or a dendritic cell or a neutrophil, comes into contact with a pathogen, they don't have any specificity. So they're going to respond the same way to any pathogen. So a macrophage is going to eat it. Doesn't matter if it's a virus or a bacteria, a parasite, whatever. The macrophage is going to eat it. Neutrophils do that too. They're actually the Marines of the immune military. They're always the first responders, like first ones in, just like the Marines, if you know the difference between some of the military arms of the military. And then dendritic cells are also considered innate because they will take in pathogen and process and present to T cells, and they're going to do that no matter what pathogen. So that's innate, and it's non-specific. Doesn't matter what pathogen it is, they have the same response. So if you are a martial artist, you would call these guys the white belts, very low level of expertise. However, some cells are specific. We call these the adaptive arm of the immune system. And the three cell types that we'll see here on this cartoon slide that are adaptive, that have specificity, are the T cells. So we have the helper T cell, the killer T cell, and then also the B cell. So T cells and B cells, just two cell types in our body that are part of the adaptive arm of the immune system. And these two cell types have specificity, meaning that they are specific for a certain pathogen, or actually more specifically, a certain portion or piece of a pathogen. We call that the antigenic determinant, or the epitope. Those are kind of like the jargon that we use in the immune world. So why is that? So I'm going to explain using this slide that I made a long time ago for my PhD defense. And I'm using it again just so that it gets a little bit more traction. So B cells and T cells are part of the adaptive arm of the immune system, and they have specificity. And they are every cell in our body. Every B cell and every T cell is really specific for only one small thing. So what I'm showing you here is this conglomeration of millions or billions of cells in our body. I, of course, only have like 100 or so on here, but this represents what we call the repertoire, the diverse repertoire of B cells and T cells in our body. And each different colored cell here has a unique or different specificity. So the way that they recognize bits of pathogen is through specialized receptors. And on the B cell, we call it the B cell receptor. And on the T cell, we call it the T cell receptor. And it's kind of a complex molecular process how these receptors are made. But the bottom line is that every B cell in our body has a unique and different specificity, and every T cell in our body has a unique and different specificity. So each cell has a unique or individual specificity, but when you combine all of them together into one gigantic pool, we have a very diverse repertoire. What this means is that anything that comes our way, any pathogen, any pathogen at all, we have specificity for it. We have probably a handful of B cells that would respond and a handful of T cells that could respond because of that unique specificity. A real life example of that would be SARS-CoV-2, the virus that causes COVID-19, was not in existence until December of 2019, right? We had cells in our body. Every one of us had cells in our immune cells in their body, B cells and T cells, that were built to recognize that. We have B cells and T cells in our body right now that are built to recognize things that don't exist, things we haven't seen. It's essentially like we have the combination for every lock already in our immune system. It just happens to be when that invader comes into our body, those cells have to be used. So that's what I'm showing you here. Every time we encounter a pathogen, only those cells that are specific will respond. So I'm showing you, pointing to this red cell. Let's say you came into contact with SARS-CoV-2. There are only going to be a few B cells in your body that could respond and a few T cells based on the unique specificity. That's the cell that's going to engage. All of the rest of the cells are going to be waiting for their pathogen. It's not their time yet. They have to wait patiently. They cannot be activated. Just the ones that have specificity for the pathogen that has invaded our body will get to play a role in the immune response. Now, don't worry, just because it's a few cells that would respond, the immune system is built for that. The cells that become activated are going to proliferate or make more and build an army of special forces, essentially, based on the recognition of the one or few cells that respond. So essentially, this should let you know that we cannot overwhelm the immune system because only those with specificity for the pathogen will respond. And it's not very many compared to the diverse repertoire that we have. Okay, so hopefully that helped. So how do we use this or that knowledge in the context of vaccines? Well, we know that the immune system, specifically B cells and T cells, can be trained. And they can study their enemy and they can create memory. So we know that vaccines train the immune system. So they act as a practice version of the pathogen to train the immune system, specifically B cells and T cells, to get ready to fight. So vaccines, we know, work with our immune system. So with the system we already have in place to provide long-lasting protection, and they also contain harmless forms of the pathogen. So harmless forms of the invader or the enemy that would be used as a practice run to stimulate an immune response and to create that memory. So the ultimate goal of a vaccine is to create memory cells, memory B and memory T, to provide long-lasting protection. Now, I'm not showing you this graph that I'm envisioning in my head, but they looked at people that had vaccinia virus, which is the vaccinia vaccine, which is the stand-in for smallpox. That's what you get vaccinated with to protect yourself from smallpox. And they found that memory B cells and memory T cells could be found 75 years later. So our memory cells stay with us for our lifetime to provide that long-lasting protection. Now, what's so important about memory cells is that these cells, as soon as they encounter pathogen, they're going to respond immediately. They don't need to become activated. There's no delay. These memory cells are going to hit the ground running. So memory cells are very quick, and they actually respond in a more robust or vigorous way as well. So they make the immune response quick and vigorous. And often our memory cells are working and work so well that we don't even know that we've been infected with something that we've been vaccinated against or something that we've encountered in the past. So memory cells are a very important portion of our immune system for that long-term protection. And then that's just a picture, an actual microscopic picture of a T cell under the microscope. Okay, quick review of the core ideas. We still have more, but this is just kind of like just to make sure we're all on the same page. So the immune system is a mobile defense system with a lot of different cells, and they work together to detect and defend us from pathogens and disease. Vaccines work with the immune system. They're harmless forms of pathogen that stimulate our immune system, and they also train the immune system, specifically B cells and T cells, to make memory for long-lasting protection. So that's kind of the core ideas from this first portion of the talk. So these are the questions we've answered. So now you've pretty much had like half of my medical school training now. What is the immune system? How does it work? What's a vaccine? How do they work? Now we're going to talk about why are there so many different types of vaccines? And then we'll also talk about why we should get vaccinated. So this is a good point. If you have any questions, please type them in the chat or the Q&A, and we'll make sure we answer those when we get there. Okay, so here are the different types or formulations of vaccines. And this is, as you can see, based on the SARS-CoV-2 virus. This is the virus that causes the disease COVID-19. So I've put these into categories. We have some formulations of vaccines that are whole pathogen, some that are parts of the pathogen, and then others that are based on genetic material or nucleic acid. So one question you might have is, why do we have so many different types or formulations? Well, the answer is that we have a lot of different types of pathogens, and vaccine biologists and scientists and physicians will work in clinical trials to figure out which formulation is going to be the best to cause the safest and most efficacious vaccine. So it really is kind of based on the pathogen. So let's talk about the whole pathogen versions. So at the top, you can see this is a live attenuated. So what does that mean? That means that the pathogen is still alive, but that it's weakened. And it's weakened to the point that it does not cause disease. So it's harmless, but it's live. From an immunologist perspective, this is actually the best version you can have because that pathogen is live but weakened. You actually have an infection and the immune system has more to play with. So your immune system is going to be your immune system will have a very wide ranging and powerful response. However, that version can't be used for every pathogen. So we have other versions as well. Another whole pathogen version is the killed or inactivated version. As you can see here, this is where it is no longer considered live, but it still molecularly resembles or looks like the whole live pathogen. This can be a very effective form of vaccine as well. However, it's very important to not destroy the virus or the bacteria to the point that it no longer looks like itself. The immune system, the cells of the immune system see or recognize the outside of the pathogen. And if the outside of this component of the vaccine, this killed version, doesn't look like or no longer molecularly resembles the wild type or the live version, then your immune system certainly will be called into play and it will make memory against it, but it will not make memory against the thing that you wanted to train it on. So it has to look enough like the original. So that can be kind of tricky, but either way, these are also very effective vaccines. So the middle category here, parts of pathogen, some of them are subunit or small pieces of the virus. And as you'll notice this here, we're looking at the outside components. So like spike protein. So with COVID, or SARS-CoV-2, this is the spike protein. That's because that's what the immune system or the cells of the immune system will see. So these just pieces, just this outer component is what you would have in that vaccine. An example of a COVID vaccine that is a subunit vaccine is the Novavax version of the vaccine. This is also true for most of the influenza vaccines, the shot form. So that would be, those would be subunits. So just pieces from the outside that have kind of just been, cut off the virus. Okay, another version for parts is toxoid. These are inactivated toxins. A lot of bacteria make toxins like diphtheria and tetanus. So these are just inactivated toxins that you would use to stimulate the immune response and train the immune system and create memory, but they're not harmful, they're inactivated. So now we'll jump to the nucleic acid based or the genetic instruction versions. And I know that you've all heard of the mRNA vaccines. So I have a couple slides here that we're going to talk about mRNA just to kind of give everyone a refresher. But it's important to know that mRNA is made off of the DNA, and mRNA codes for, or it's the instructions to make protein. So what vaccine biologists did was they figured out the section of the genetic sequence of SARS-CoV-2 that coded for the spike protein. They isolated that bit of the RNA, and then they packed it into this kind of like lipid or fatty shell to deliver it. So that's all that's in that nucleic acid-based mRNA vaccine. You can think of that, the lipid shell is like a suitcase to carry the mRNA into our body and into our cells. Turns out that our cells are made up of phospholipid bilayers. So this actually works seamlessly to get into our cells. And then we have the DNA, DNA in a vector down here. This would be like an example of the J&J vaccine, the Johnson & Johnson. This is just a step up from mRNA. It's the DNA, so the genetic instructions, that's DNA, which would then, you can make mRNA or messenger RNA off of the DNA, which then codes for the instructions to make the protein. So I'm going to give you an analogy. The DNA is the cookbook, the mRNA is the recipe, and then the spike is the protein that's made from the recipe. So we have the cookbook, which is the DNA. We have the mRNA, which is essentially the recipe for the protein, and then it makes whatever that recipe is, and in this case, it's making the spike protein. So either way, these mRNA vaccines or DNA vaccines result in the production of spike protein. Okay, so those are the different formulations. Now I just want to go through DNA versus mRNA so you can have a totally clear understanding of how these vaccines work, because I know that these are the most commonly asked questions that I get. So this is every cell in our body. This is a cartoon that represents every single cell in our body. In the nucleus, which is the center of the cell, lives the DNA. So that's the genetics instructions for everything that's needed. And every cell in our body has the same DNA. It's how it's used. So different cell types use different parts of the genome to become like a lung cell or a heart cell or a kidney cell or an immune cell. So they use different pieces. So the DNA, again, is the cookbook. If your cell needs to make a certain protein, it is going to transcribe or make a piece of RNA from the DNA, just a little piece, just a tiny bit. So the cookbook, DNA, one of the recipes is the RNA. In order for that to be read by the cellular machinery that makes proteins, it has to be a messenger RNA. So you can see that here. That messenger RNA then leaves the nucleus and goes into the cytoplasm where the cellular machinery to make protein lives, and the protein is made in the cytoplasm. This is true of every cell in our body. And this process of making protein off of mRNA happens every day, all day long, in every cell of our body. So this is something that is naturally occurring every day, all day long. And even though the machinery to make protein is invisible in this cartoon, it just reads mRNA all day long, all day long, all day long. And mRNA has a very short life because we only need proteins for a certain period of time. Our cells don't need to make every protein all day long. They'll need to make a certain protein for a certain time period, maybe a certain 5 minutes after you ate, or different timing. So mRNA is very volatile in the sense that it is degraded quickly. So that will be key here in just a second. Okay, so that's how we call it the central dogma of molecular biology, DNA to RNA to protein. So When vaccine biologists were trying to come up with a vaccine for SARS-CoV-2, they used this technology, which had been under research for at least 20 years, but hadn't gone to clinic yet. But they're using this particular type of vaccine formulation. They studied the coronavirus. They figured out the genetic code, which piece of mRNA coded for the spike protein. And then that was used in the vaccine to allow the cells of our body to use this everyday process to make the spike protein. Spike protein would then be detected as foreign by our own bodies, and the immune system would respond and make the antibody and make the memory B cells and T cells. So it just was working with our immune system in ways that we know how it works. This is a slide or a photo from a paper in Nature from April 2020, just to give you a little bit of an idea of how this works. So here's the RNA, mRNA that codes for spike protein in that kind of lipid shell or suitcase. It's injected into your arm usually. That gets taken into the cell. The cellular machinery makes the spike protein off of the messenger RNA. That spike is foreign. It gets picked up by those. This is the dendritic cell. That's the sentinel I told you about earlier. It's going to process and present, show it off to T cells, and an immune response ensues. So we're going to have B cells alerted to make antibody, and memory B and T cells will be created as well for long-lasting protection. And then, like I told you, mRNA is very short-lived. So, fun fact, if you look at the data from Moderna and Pfizer's clinical trials, the mRNA from the vaccine is gone within 24 hours of injection. So if anyone out there Snapchats, that's a message that teenagers use. It's a social platform that, like a chatting platform that teenagers use, where you can send a message and it like magically disappears in 24 hours. That is what the mRNA does as well. It's gone in 24 hours. Because again, mRNA is used every day, all day long. We don't need never-ending amounts of protein, so mRNA is degraded very quickly. So you can think of it as a Snapchat message. Okay, so back to the different types of vaccines. I just wanted to show you, again, we have all these different versions, and they're driven, the formulation we choose is driven by the pathogen, as well as looking at the efficacy and safety in clinical trials. Okay, now vaccine options right now for SARS-CoV-2 or COVID-19 are listed here. So we have parts of a pathogen like we see in Novavax, and that's just the outer spike protein that's in the vaccine, nothing else. We have the Moderna and Pfizer nucleic acid-based mRNA vaccines that are in those lipid or fatty shells. And then we have the AstraZeneca or the J&J version of the DNA for the spike protein that's in a vector. So those are the different vaccine options for SARS-CoV-2. So we can look really quickly at their immune response, the immune response to the vaccine. So if you look here, this is showing us SARS-CoV-2, which is just simply representing the different versions of the vaccine. Again, these vaccines are not using the entire virus. So if we look at the Novavax first, again, that's just the purified spike proteins, just that outer piece of the virus that is being injected. Sentinels, macrophages, dendritic cells will pick that up, show it off to T cells. B cells will also be alerted, will have an immune response. We're going to have antibody produced that will remain in our blood, and we'll have memory B cells and T cells created. So they're going to be trained and ready to fight. Second version we have here in the middle, we have the DNA version of the vaccines, the DNA vector. Again, Johnson & Johnson or AstraZeneca. That DNA is going to be in a viral carrier that's injected into the body. The DNA is transcribed into messenger RNA. Messenger RNA is read by the cellular machinery to make the spike protein. Spike protein is picked up by those sentinels. degraded and processed and presented and put onto the surface and shown off to B cells and T cells. And here we have antibody production and then memory B and T cells created. And then finally with the Moderna or Pfizer versions, again, not the DNA version, but messenger RNA. So that's the message itself that codes for the spike protein. That's what's delivered. Our cellular machinery makes the protein right away. That's going to be picked up as foreign by the sentinels, again, like macrophages, dendritic cells, and an immune response will ensue in which antibody is produced. And we have memory B cells and T cells. So they all kind of go, even though there's differences between them, they all result in the production of antibody. Those, again, are the arrows being flung by the B cell archers into the blood. And then we have memory B cells and T cells created. Okay. So now that you know all about that, I like to come up with analogies, as you've probably noticed, for everything I teach, but for vaccines in particular. This is another graphic design, homemade. So vaccines for COVID-19 are like seatbelts. So whenever you get into a car, you put on your seatbelt. You know that it's not going to necessarily prevent you from crashing into another car, but you do know that that seatbelt is going to protect you from likely being hospitalized if you are in a crash, likely will prevent you from serious injury, and in most cases will also prevent death. So this is how the COVID-19 vaccines work. They protect us, maybe not from the crash, but decreasing hospitalization, preventing serious injury, and preventing death. So they're like seatbelts. Okay, so we've learned now that multiple types or formulations of vaccines exist because there are multiple forms of pathogens. And the best and most effective form will be selected by vaccine biologists as they are undergoing vaccine development. So let's learn a little bit more about why should we get vaccinated. Well, there are lots of reasons. The first one is that you might want to protect yourself. So vaccines will train the immune system to create memory and provide that long-lasting protection. And they're going to prevent us from becoming sick. So protect us. Another reason is you might want to get vaccinated to protect other people. We know that there are some people in our community who can't be immunized due to health issues or the status of the immune system. So some of those people might have a weakened immune system. They might have an immunodeficiency. They might be undergoing cancer therapy like chemotherapy. Maybe they have had a transplant, so they're on immunosuppressives. And these people need our protection. Infants are too young for some vaccines through clinical trials, looking at when infants or young children respond most effectively. It's not always at birth. So infants are too young for some vaccines. So we need to protect those young children and infants. And then people who are pregnant also can't get all of the vaccines. There are some that are recommended during pregnancy, like influenza or Tdap, and also the mRNA COVID vaccines are recommended because mom would produce antibody that crosses the placenta that goes into baby to provide protection. But not all vaccines can be safely given to pregnant women, particularly those that are live.
Dr. Bernard, we have a question in the Q&A.
Sure. Yeah.
It says, I'm going to say it wrong, so forgive me. Dendritic cells actually mobile? Yes.
They are all mobile. All of them move. They are all capable of moving. Yep, they move. Great question. They sometimes will, once they show off their stuff, what they've found to T cells, they might kind of sit there for a while, but yeah, they are mobile. They move. All of the cells move around our body. It's like this amazing mobile defense system. So yeah, great question. Okay, I wanted to talk to you a little bit about what's called herd immunity or community immunity. And that's where if enough of us are vaccinated, we will shield those who are not vaccinated for whatever reason and decrease the spread of disease. So it's important that we have certain levels of vaccination to keep us all healthy, particularly those that have conditions where they cannot be vaccinated. So I'm going to go through this kind of cartoon with you about herd or community immunity. And I want to draw your attention to the top left. Here in red, we have individuals who are diseased, so they have the disease. In blue, those are individuals that are susceptible, so not vaccinated. And then in gold, because if you're vaccinated, you get a gold star. Those that are in gold are vaccinated. So this first scenario we're going to look at, there's no vaccination. So this could be thought of as like when COVID-19 started, no vaccination, no protection whatsoever. If you have one infected individual and the rest are not vaccinated, that's going to spread very quickly like wildfire through the population, and you're going to see vast amounts of infection. Second scenario is where we have vaccination below the threshold for protection. It's important to know that we have different thresholds that are necessary for different vaccines, and that's really based on the pathogen itself. How contagious is it? How quickly does it spread? Like as an example, measles is still probably the most contagious pathogen that we've encountered. We need 95% of the community to be vaccinated to truly provide protection. Anything below that number, we're not going to protect everybody. And different pathogens have different thresholds. Some are slightly lower. I think on average, we could probably think of about 90% as a good threshold for just about anything. That would be ideal. But in this one here, we have vaccination below threshold for protection. So if you look at the one diseased individual, you have roughly half vaccinated, half not. That's going to spread through the unvaccinated population pretty thoroughly. Now the vaccinated will be kept or protected, but those that are not will definitely get infected and get disease. On the right here is best case scenario. Vaccination is above threshold for protection. You have one infected individual and you have the majority vaccinated. Those that cannot be vaccinated are shielded by those that are vaccinated. So you're going to have the infection will not spread through that community. So this is ideal state here. Okay, so we get vaccinated to protect ourselves from disease and also to shield our community from the spread of disease. So we have covered all of the questions. You guys could now teach this for me, probably. So with that, I know, let's see, we're at time. You can ask questions now if you'd like, or you can email them later. And then I also wanted to show you where I got some of my info. And then if you wanted to listen to my podcast or look at my TikToks, I'm at Immuninja, and I also have an Immunology 101 series blog. So with that, I'm done. Any questions?
I see one here in the chat asking about the recording, and we will be sending this out to event attendees after this. And then we also put it up on the CU Anschutz alumni YouTube channel. So we'll also put a link to that as well.
Awesome. That's all the questions. It was crystal clear.
There's one that says, so what is the bad news?
The bad news is that we're not all vaccinated. And the other bit of bad news, I think, is that SARS-CoV-2 keeps changing. Just like the influenza changes every year and we need to have a new annual flu vaccine, SARS-CoV-2 seems to be very, very good at changing. So we hear about all these variants all the time, which just means we're going to have to continue to update and likely have an annual vaccination for SARS-CoV-2. So yeah.
Keeps going, huh? Then we have a few more questions in here.
Yeah, sure.
They're coming in. What is an immune storm and how does it occur?
Okay, so I think what you're referring to is something called a cytokine storm. So a cytokine storm is when your immune system, particularly T cells of the immune system, will when they're activated, they'll secrete a large amount of chemical mediators called cytokines. And cytokines coordinate and regulate immune responses. And different cytokines do different things, but ultimately they will kind of enhance the immune response. And if you have too many cytokines released, you can have what's called a cytokine storm. That can result in like a decrease in blood pressure and shock and death. But this usually this would not happen like after a vaccine. As an example, there is a immunotherapy called CAR T cells, or chimeric antigen receptor T cells, that are given to some cancer patients. And they found sometimes that when they injected these early on, that would sometimes cytokine storm. So because of that, they very closely monitor cytokines in the blood, and they can counter those effects with interestingly some like cytokine kind of blockers. So that's the cytokine storm.
I have never heard of that. Thank you for sharing.
Yeah.
Some more questions in here. How do you decide when boosters are needed?
So this is usually, so I'm just going to tell you right now, and I know that you are using the common terminology. So but I don't like the word booster, because people always think booster means, oh, it's my last shot ever. I like to use the term additional dose because we will always need additional doses. But the way that that's decided is if in clinical trials, there would be, like for some of the vaccine series, we have a series of boosters that are needed. And that's because in clinical trials, they will have shown that they're not as effective with just like 1 or 2 shots. You would need 3 to complete the series. In the context of COVID, it's because what we have found by looking at people's response to the vaccines is that antibody, that the arrows in the blood that really kind of neutralize and surround the virus when it enters our body, we need a lot of that in the blood for that to work really well. We found with these COVID-19 or SARS-CoV-2 vaccines is that antibody levels after about 6 to 9 months start to wane or decline in response to SARS-CoV-2. So in order to keep those antibodies levels high, you want to have an additional dose to kind of reinvigorate the immune system, get those B cell archers to make more antibody. So you do have memory. You still have memory B cells, you have memory T cells, but it's the antibody that's just not hanging around. So that's why we get those additional doses for COVID.
Okay. There's a question here on new COVID vaccine versus the older versions.
Yeah, so the difference really is just the variants that they're against. So we had the first set of COVID vaccines were against like the original strain or variant. Believe it or not, the term variant and strain mean different things to virologists. So we're going to use the term variant because that's actually correct. Variants. So when you're getting original as well as one of the Omicron variants. So that's the difference between them. We do find that your immune response to the original can still provide pretty decent protection against the newer variants because they do look similar. But if you can get the newer vaccines against the newer variants, that's just going to provide additional training and better protection.
Thank you.
Yeah.
There's a question here of, can you discuss the difference in sensitivity, specificity in mRNA, DNA, and Novavax?
Yeah, so the mRNA vaccines and the DNA vaccines give a really, I think, well, first of all, let's just say that they've all shown to be safe and effective. Otherwise, they wouldn't be out there. Vaccine biologists, in all these clinical trials, if it's not safe and it's not effective, then we just trash it and, it wouldn't have gone on. So the mRNA and the DNA vaccines give a very high level of protection. Novavax vaccines, that's the subunit, just the pieces of spike protein that's in there. The reason why that one's probably just a little bit less, it's still effective, but maybe it doesn't provide as great of protection or as high level, is because the mRNA and DNA vaccines allow the machinery of your cells to make spike protein and make a lot of it in a short period of time. The Novavax vaccine, it's the subunit. What you get in the vaccine is all your immune system gets to play with. So your immune system, or your cells actually make more spike protein if they're given the recipe than what's in the Novavax vaccine. But I would say, if you were to go to your pharmacist or your doctor and they said, we have Novavax or nothing, go with Novavax. Higher than nothing. So I hope that helps.
No, thank you. That's amazing. The next question is and you kind of touched on this already, but I just want to make sure we answer the specific question. With all the mutations and variances, do we know the ideal timing of COVID-19 boosters, or we'll say additional doses?
Additional doses, yes.
I'm learning tonight.
Yeah. So I would say that, they're trying to right now to put it on an annual schedule so that it will coincide with the annual flu vaccine in the fall. Is that ideal from an immunologist or just a public health perspective? Probably not 100%, because these variants are going to come out. They come out at different times. We can't predict when these different variants come out, but in order to keep the population, most people don't want to. We've found that not everyone, as soon as vaccines are, announced, not everyone just runs to their doctor. So we're trying to make sure that compliance or getting the vaccine is as convenient as possible. And having an annual shot aligned with another annual shot is likely the best way to do that. Does it mean that we might miss a window of time for certain variants? Maybe, because we don't see SARS-CoV-2 as being seasonal. Like the flu is totally seasonal. We've worked that out. It comes like you should get your shot probably late September, early October. You've got it wired. COVID-19, not exactly. So it's not seasonal like that, but having this is more for convenience and making sure that everyone gets it. So that's really kind of the ideal timing for that additional dose. However, I think if you are immunocompromised or above a certain age range, like 65 and older, that if there is some new vaccine that comes out, before the window of time to have it be given to the most of the population, that those people can likely get that vaccine right away if requested.
If you're okay, we have 3 more questions and then we'll
Sure.
end. Okay, great. So the next one is, why do reputable immunologists question the effectiveness of vaccination?
Oh, this is a loaded question.
I thought you know this one.
Well, I'm not sure. Okay, the word reputable. Let's see. So first of all, I'm going to tell you guys something that probably would surprise you, but I think that everyone should question and be skeptical of anything you put in your body. You should probably learn how it works. And this includes alcohol, fatty foods, Doritos, whatever it is. If you're going to if you're going to question what goes in your body, you should question all of what goes in your body. But I think it's important for people to understand how they work and have questions about them. Now, people who are looking at the data and who are just very immersed in the world of immunology and vaccine biology, we do not question the effectiveness of vaccines that have evidence to prove that. There are some people that might question it before they read that, or others that might question it and not actually dig into the data. But I don't think that anyone who is an actual immunologist that's looked into the data and has asked the questions and had really, good discussions with other experts in the field would question the effectiveness. I hope that helps. You're welcome to email me if you'd like.
Thank you for that. The next one, this is interesting, is global warming changing our immune system? And it says too much hygiene.
I don't know that global warming or climate change is changing our immune system. And the too much hygiene, that's called the hygiene hypothesis. Those are a little bit different. But I will say that in the context of climate change, what we're noticing is that certain pathogens, like particularly pathogens that are carried by like mosquitoes and things like that, are moving into territories they never used to live in. Like yellow fever is considered something that's more South America. Well, as climate change occurs and, more northern areas are going to start to increase their temperature, we're probably going to see yellow fever and malaria and things like that move with the warmth, because that's where these things live. So I would say it's going to change where pathogens can be found. It's not really going to change how our immune system works. But then the hygiene hypothesis is something different. That is where the idea is that people that live in conditions where they're too clean, like they use Purell all the time, and they wipe down everything, and you're not exposing your immune system to like enough stuff. Some studies have shown that people that are overly hygienic, that they're more susceptible to things like allergies and maybe autoimmune disease. However, I will say that has also been called into question, and that there are some studies starting to come out saying that there's not as big of a link to that as originally thought. So bottom line is, if you don't expose your immune system to anything, it is not going to have memory. It's not going to have antibody. You really do want to send your kids outside to play, to expose themselves to stuff and build that memory. So I hope that helps.
Thank you. My children have had plenty of exposure to all the things.
There you go.
So our last question here tonight is, which of the modalities of vaccination was the most effective for SARS-CoV-2?
Yeah, so I think that all of the vaccines that have been given approval have been effective. I think that if you were to look at the data, it's probably the mRNA vaccines would probably have the highest. It's like, in the 90s, 93, 95 percent protection. The DNA vaccines, I think, are up there pretty high as well. Novavax, I think, is lower, like maybe mid to high 80s. Again, it's the subunit. Your immune system only gets to play with what's in the vial, the syringe. But either way, they're all those that are out and approved or under EUA, emergency use authorization, are quite effective. I mean, mid to high 80s is still really good. I mean, if you look at some of the annual influenza, and that, again, we can only work with what we have for pathogen, respond to every pathogen really vigorously. So you can only, that's a whole nother kind of topic. But like the annual flu vaccine, we get really excited with like 70%, so these SARS-CoV-2 or COVID vaccines are, off the charts, like, wow, this is great. So why does the antibody level decline? We don't know. If we did, we would fix it. But that's something that I think has to do with just the pathogen itself and how our immune system across the board just responds to it. So those are things we have to work on. Immunology, job security. And fascinating, but I'm not biased.
Well, thank you so much. I'm sure I speak for everyone. I'm so grateful that you would share your expertise, and to have this knowledge on our campus is amazing. So we appreciate it so much. Before we close tonight, I do want to share a couple pieces of information that we have for you. So our office is partnering with the CU College of Nursing and the Office of Continuing Education at the CU College of Nursing on an upcoming Cultural Considerations in Healthcare virtual lecture series. So this series is offered virtually during the summer, as you can see here for the dates, and there is a discount for all alumni of the campus. There are NCPD credits available through nursing if that's something of interest to you. So we'll send more information out on that today. And then, post-event. And then next we have some upcoming events we just wanted to make you aware of. So the CU Nursing Alumni Association is hosting brunch at the Denver Museum of Nature and Science on July 15th. And we also have CU at the Rockies. So if you want to come to a Rockies game, a ticket purchase gets you a CU themed Rockies hat. And there is a pre-reception at Jackson's. And then we'll ask you to save the date for our all alumni celebration coming up on September 29th and 30th. That will have Dr. Terry Fry talking about the amazing work at the Gates Institute, afternoon at the zoo, campus tours, a happy hour at Pindustry. So we have a lot of fun activities happening that weekend. So thank you so much for joining us this evening. Thank you again to Dr. Bernard for sharing your expertise, and we hope you have a great evening.
Thanks for having me.